Hello everyone. So we have a high yield concept that appear repeatedly in the exam and has multiple clinical correlation. A lot of disease is associated with this topic, which is collagen synthesis. So I will explain us a brief review of steps of collagen synthesis and the disorder associated with defect of each step. So basically we have here the nucleus, and we have the cell, and we have the nucleus and the rough endoplasmic reticulum. The site where the beginning of the collagen synthesis is in the rough endoplasmic reticulum in the cytoplasm. There is no step in the nucleus. So in the rough endoplasmic reticulum, the mRNA collagen will form a peptide collagen as a general role for the protein synthesis. We have mRNA, and we take the glycine peptide and added it to either proline or lysine. So the final protein is XY. So why XY? Because XY is either proline or lysine. So in this form, it will form as this glycine XY. So we form a glycine, proline and lysine. This is called the pre-pro collagen. Okay. There is a old question that asks about which is abundant amino acid in collagen, and the answer is glycine, because glycine form 1/3 of the collagen. The other is the proline and lysine. So pre-pro collagen undergo many steps. The first step is hydroxylation. This is hydroxylation, the first step. Hydroxylation means added OH group to the pre-pro collagen. This also a high yield steps because it require vitamin C. So we form by hydroxylation the glycine, proline and lysine undergo hydroxylation. Defect in the hydroxylation will lead to scurvy. Okay, so the question asks, if the patient is vitamin D deficient, he will be unable to hydroxylate the collagen and lead to collagen synthesis disorder. So presented as petechia, hemorrhage, and the other sign and symptom of scurvy, such as gingival bleeding when a patient brushes his teeth and so on, due to defect of vitamin C and defective hydroxylation of the collagen. The next step is glycosylation. We added the glucose group on the OH. We will form a pro-alpha chain. This is a pro-alpha chain. This is pro-alpha chain. Okay, a pro-alpha chain also combined together with another two pro-alpha chain by hydroxyl bond and disulfide bond to form a triple helix. This is a triple helix formation. Triple helix formation, or also called the pro-collagen. This is the same as triple helix. It form of three pro-alpha collagen. So a three or a triple pro-alpha chain. So defect in the triple helix formation lead to osteogenesis imperfecta. As you know, the osteogenesis imperfecta is also a defect in collagen 1. So triple helix formation defect lead to osteogenesis imperfecta. The child presented with the blue sclera and multiple fracture, recurrent fracture, and so on. So we now form the triple helix or pro-collagen. The next step is exocytosis to the extracellular space. All the previous steps is occur in the cytoplasm or in the rough endoplasmic reticulum, the same. And also I will mention that the no step in the collagen synthesis occur in the nucleus. This is a wrong answer on the UOLD or on BME. So after exocytosis of the triple helix or procollagen, it will undergo enzyme cleavage. The next step is enzyme cleavage at the end that rich in disulfide bond. So this is the end that rich in disulfide bond by transpeptidase collagen enzyme. So this is also asked in the UWorld. The defect in the enzyme cleavage of the procollagen or triple helix lead to Ehlers-Danlos syndrome. Okay, Ehlers-Danlos syndrome. Also, after formation of the enzyme cleavage, we will have another structure called the tropocollagen. This is a tropocollagen. This one. So the tropocollagen will also bind to another tropocollagen by the enzyme is called lysyl oxidase enzyme. This enzyme depends on copper. So in Menkes disease, when we have a defective in copper absorption, this step will be defect. So we cannot cross-link the tropocollagen to one another because a copper deficiency. So the lysyl oxidase enzyme activity will be low. So we will only get a tropocollagen. We cannot cross-link between them. So after we cross-link the tropocollagen, these three tropocollagen, by a bond between them, this is the bond between them, this is tropocollagen, we will have finally the collagen fibrils. So this is collagen fibrils. After we cross-link the tropocollagen, this is tropocollagen, we cross-linked each other by the lysyl oxidase enzyme. So a quick recap: if we have a defect in hydroxylation which require vitamin C, we will have scurvy or vitamin D deficiency. If we have a defect in the triple helix formation, we will have osteogenesis imperfecta, which is also a defect in collagen 1. Also, if we have a defect in enzyme cleavage of triple helix at the disulfide bond, we will have Ehlers-Danlos syndrome. If we have a defect in cross-link of tropocollagen, we will not form the collagen fibrils, which is occur in Menkes disease. Okay, so defect in tropocollagen cross-link occur in Menkes disease. Also, I should mention that the collagen, after we found collagen, we have 4 types of collagen: collagen 1, collagen 2, collagen 3, and collagen 4. A quick mnemonic to know which type occur in which organ is scab. Type 1 occur in S skeleton, which include bones, dentin, fascia, also scar. This is C, S, skeleton. C, this is in cartilage, type 2 in cartilage. And also nucleus pulposus of the intervertebral disc of the spinal cord. A, type 3 in arteries. And also in the retina. And the B, type 4 in the basement membrane. So type 1 collagen, if we have a defect in type 1 collagen, we will have an osteogenesis imperfecta. In type 2 collagen, we don't have a specific disease. In type 3, we will have Ehlers-Danlos syndrome, vascular type. And if we have a defect in collagen 4 in the basement membrane, we have either defective collagen 4, which occur in Albert syndrome, or we have antibody against the type 4 collagen. This will lead to good posture syndrome. And that's it for the collagen synthesis disorder and the steps of the collagen synthesis. Thanks you for listening.